Hello students, welcome back. In this video, we'll be learning about the last topic of the chapter 5, fundamental unit of life, uh, regarding cytoplasm. Cytoplasm, the content of a cell except for the nucleus. It includes cytosol, the fluid, organelles, vesicles, and the cytoskeleton. Cytoplasm has two parts, cytosol and cell organelles. Cytosol, it's a fluid part of cytoplasm, homogeneous and crystallocolloid liquid. Crystallocolloid, what is the meaning of crystallocolloid? It is crystals and the colloid, that is suspension. That contains substances like water, ions, enzyme, vitamins, amino acid, carbohydrates, lipids, proteins, etc. Cell organelles. What are cell organelles? These are the submicroscopic structures which are specialized to perform specific cellular functions. Cell organelles may be bounded by a membrane, as in eukaryotes, but prokaryotes like membrane-bound organelles. So the membrane-bound organelles that we will be discussing here are absent in prokaryotic cells. Viruses lack cytoplasm as well as the membrane. They don't have any cytoplasm. They don't have any nuclear envelope. Only what they have the genetic material and the outer protein coat. That's it, right? And they are known as the connecting link between living and non-living. Moving ahead. Now we will discuss about the cell organelles one by one. The first cell organelle is endoplasmic reticulum. The endoplasmic reticulum is a network of interconnecting membranes distributed throughout the cytoplasm. The internal compartment, called the lumen, is a separated part of the cell with a distinct proteins and ion composition. The endoplasmic reticulum folding generates a surface area much greater than compared to the plasma membrane. Depending on the presence and absence on the membrane of the presence of the ribosome on the surface, there are two types of endoplasmic reticulum. One is rough endoplasmic reticulum, another one is smooth endoplasmic reticulum. Here in the diagram, it will be clear to you. This one is the interlumen, that is a space, right? My dear students, in bio, again, these words, whenever it comes lumen, that indicates the inner space, right? As you can see, it is highly coiled, right? The membrane, if they have the ribosome, are known as rough endoplasmic reticulum, and the one which doesn't have ribosome on the surface are known as smooth because of their appearance. What is the function and the role of ER? Protein synthesis takes place on rough endoplasmic reticulum. Lipid synthesis takes place on by smooth endoplasmic reticulum. Proteins and lipids are synthesized on ER are used for making cell membrane. My dear students, when you will read the chapter from the textbook, you'll come to know about the one word, that is membrane biogenesis. That is, membrane is the plasma membrane, bio is the living, and genesis means to produce. That is, for the production of membrane, endoplasmic reticulum is essential. Proteins and lipids are synthesized by endoplasmic reticulum that are being used for the synthesis of membrane, and that term is known as membrane biogenesis. What else are the functions of the ER? That is, transport. It helps in intercellular and intercellular transport of substances. It gives internal support to the cytoplasm. SCR, that is smooth endoplasmic reticulum, is present in liver that take part in detoxifying many poisons and the drugs. Right? That's why it is known as, liver is known as a graveyard. Whatever things the rat cells, they first go over there, the antibiotic, the drugs, and yeah, specifically the drunkards, their liver get damaged. Why? Because they drink more and more alcohol that goes inside the liver for detoxification, and that leads to damaging their liver, right? So why it is so? Why liver is a graveyard? Why it helps in detoxifying? Because of the presence of more number of smooth endoplasmic reticulum. Now let's see the difference between smooth endoplasmic reticulum and the rough endoplasmic reticulum. Smooth endoplasmic reticulum ribosomes are absent in them. It synthesizes lipids and steroids mainly formed of vesicles and tubules. Whereas on the other hand, rough endoplasmic reticulum, ribosomes are present, synthesize protein and formed of cisternae. Right? Next is Golgi complex. Golgi complex is an organelle of a membrane-bound sacs, tubules, and vesicles. It's not like, my dear student, it's not like that endoplasmic reticulum continue. Here you can see the vesicles. The tubules are lying one on, like one after other. Vesicles secrete complex biochemical. Golgi is a simple complex in animal cell. In animal cells, it is present in the form of Golgi, whereas it is in plant cell, they are formed as a unit and they are known as dictosome. Membrane bound sacs of Golgis are called cisternae. Cisternae occur in stacks, tubes, and vesicles occur on their periphery. Here you can see in the diagram, this is endoplasmic reticulum and here is the Golgi. These are the cisternae, right? And actually it has two phases, the C, that is cis phase, and the opposite of cis phase is the trans region. Moving ahead, the function of Golgi apparatus. Golgi apparatus is involved in repair and synthesis of cell membrane. Lysosomes are formed by Golgi apparatus. All types of substances which are to be secreted or excreted are packed in vesicles of Golgi apparatus. So thus, my dear students, the main function of Golgi apparatus is storage, modification, and packaging of the substances, right? Next comes the lysosomes. Lysosomes are generally found in animal cell. They lack in plant cell. They are not present in plant cell. Are small spherical vesicles covered by single membrane contain digestive enzyme, right? What is their function actually? The function of lysosomes are, they help in destruction of foreign particles. If our cell get damaged, or if they help in removal of worn out cells, they help in removal of the substances that are harmful to us, right, to the cell, or even they damages, if our cell is not functioning or it's damaged by some pathogens, then they by themselves burst and kill entire cell. That's why they are known as suicidal bags. They help in the removal of cell debris during the time of metamorphosis, that is change from young to adult, larval stage to adult. The organs are digested by lysosome. And where it is lysosome is being synthesized, it is synthesized in Golgi. It is packed in Golgi apparatus, right? But the enzymes are synthesized in rough endoplasmic reticulum. Next organelle is mitochondria, which is known as powerhouse of the cell. Mitochondria is a rod-shaped cell organelle of aerobic eukaryotes which take part in respiration. Why, my dear students, it is known as powerhouse of the cell? Because mitochondria is responsible for the synthesis of energy during the time of respiration in the form of ATP. Right? Why it is able to produce energy? Because of the presence of enzyme. It is double membrane organelle, the outer membrane and the inner membrane. The outer membrane is smooth and the inner membrane is highly folded and porous. Right? The cristae, that are the folds inside, are known as the cristae that contain the enzyme that take place in respiration. Right? Mitochondria has its own DNA, ribosome, and enzyme. That's why the mitochondria is also known as semi-autonomous organelle. Many of the time in the exams also it is being asked the name the semi-autonomous organelles. So the answers are mitochondria and plastid. Why it is known as semi-autonomous? Because it has its own DNA. It has its own ribosomes. So it is able to synthesize its protein and enzyme, right? This is the diagram of mitochondria. The matrix, that is inner membrane. Let's begin with the outer membrane. Inner membrane, which is highly folded, and these folds are known as cristae. The inner folds are known as the cristae, and the inner membrane is known as matrix. Moving ahead, the plastids. Plastids are only present in plant cell. There are three types of plastids: leucoplast, chromoplast, and chloroplast. Right? Leucoplasts, they are colorless. Chromo are the non-green, colorful, and chloro are the green. What is the role of leucoplasts? Leucoplasts are the colorless, cylindrical or the round, cylindrical to round in shape. They are found in exposed cells. They take part in storage of food and can change to other types as well. Chromoplasts are of different colors. They are irregular in shape. In exposed and unexposed cells, they are present in all the kind of cells. The colorful part, as you can see, chromoblasts are present in petals, flowers. The colorful part of the plant is because of the presence of chromoplast, the colorful part, right? Next is the chloroplast, which is the important one. Chloroplast is known as kitchen of the cell. It is bounded by two membranes. My dear students, plastids and mitochondria, the similarity is one, they contain their own DNA and ribosomes. Second similarity is they both have double membrane. They have two membranes. Endoplasmic reticulum, lysosome, and Golgi apparatus, they have single membrane, whereas mitochondria and plastids have two membranes, right? Inside of chloroplast is clearly marked into a colorless matrix called stroma, right? And a membranous system called grana. Each grana consists of a stalk of membrane-bound flattened sac called thylakoids. Thylakoids contain the pigment chlorophyll. Stroma contain enzyme for the dark reaction. Now, what is dark reaction? That is a part of photosynthesis. As I told you that the chloroplast contains its own DNA, RNA. That's why this is also known as semi-autonomous organelle. Here you can see in this diagram the two membranes and the plain matrix is a stroma. The coin-like structures are known as thylakoid and the stack is known as granum, right? What is the function of plastids? Plastids. Chromoplast provides color, as I told you. It is present in fruits, flower that attracts animals to help in pollination and fruit disposal. Leucoplasts take part in storage of protein, starch, and oil, that is food. And chloroplast trap solar energy and help in synthesis of food by the process known as photosynthesis. Next, ribosomes. Ribosomes are known as protein factory, right? They are responsible for the synthesis of protein. Ribosomes are the unit no membrane organelles, right? They are composed of RNA and proteins. There are two types of ribosomes, that is 70S and 80S. 70S is present in prokaryotes and 80S is present in eukaryotes. Next is centrosome, that is central apparatus. My dear students, centrosome is only present in animal cell, which is absent in plant cell. It is a small naked organelle present near the nucleus in the cell of animals and some lower plants. It has sheath of special cytoplasm. You can see it has a cartwheel-like structure, right? It encloses two microcylinders called centriole. Two centriole-like right angles. Now, this is a structure. You can see a cylinder-like structure which is forming a cartwheel. What is the role of this centrosome? They form the astral spindle fibers during the time of DNA. When the nucleus, the cell is dividing, during that time, it forms a fiber that are helpful in segregation of chromosomes, or else they are also present in the cilia and flagella of the lower organism. Vacuoles. Vacuoles are the storehouse of the cell, membrane-bound, non-cytoplasmic sac that contain non-living solid or liquid content. They are known as the storehouse of the cell, right? They are of three types: sap vacuole, food vacuole, and contractile vacuole. Covering membrane of the sap vacuole is known as tonoplast. The fluid-filled content of the vacuole is known as sap or the tonoplasm. Recently, digestive enzymes are reported in vacuole of plant cell. This is the recent discovery in which it's been said that vacuoles contain the digestive enzymes as well. Dear students, the vacuole, here you can see a vacuole. This is the plant cell. If you will observe the plant cell, it has the larger vacuole comparing to animal cell. Animal cell may or may not contain the vacuole. If it's present, it is in smaller many. Difference between plant cell and animal cell. Plant cell. Plant cell have cell wall is present in them, plastids are present, centrioles are absent, vacuole is large and less. Nucleus lie on one side, cell is rigid because of presence of cell wall. Golgi contain dictosome, cell is larger in size, starch is stored. Whereas on the other hand, animal cell wall is absent, plastid is absent, centrioles are present, vacuoles are small and many. Nucleus lies in the center. Cell is flexible because they don't have any cell wall. They have plasma membrane of the cell membrane in the form of vesicle. cell is smaller in size, glycogen is stored. Here, one more diagram to differentiate between the plant and the animal cell. You can see, let's begin with the plant cell. The centriole is present here, centriole is absent here. Right? Chloroplast is present here, which is absent in here. Rest all plasma membrane, lysosome, Golgi apparatus, SER, nucleus, RER, ribosome, mitochondria, peroxisome, cytoskeleton are present here. If we compare about the vacuoles, vacuole here is large, whereas here vacuoles are absent, right? And the cell membrane of the vacuole is known as sternoplasm. The last topic of the chapter is types of cell division. Cell divide, right? Yes. From one cell to other, from the pre-existing cell, the new cell arises. There are of two types of cell division. Number one is mitosis and number two is meiosis. Mitosis. Here, my dear friends, you can see if there are 4 number of chromosomes, then they are equally distributed in daughter cells, right? Whereas here, the 4 are divided into two in each, right? And the product here in mitosis, you will find 2 daughter cells, and in meiosis, you will find 4 daughter cells, right? Now let's see the difference between mitosis and meiosis. Mitosis produces genetically identical cell, right? Results in diploid cell. Diploid cell, that means same number of chromosomes, right? Take place throughout an organism lifetime. Whenever you are getting any wound or any cut, the new cells arises. It's because of the result of mitosis. It involves an asexual reproduction. Whereas on the other hand, meiosis produces genetically unique cells. Results in haploid, that means half the number. As I told you, in mitosis, you can count. It has 4 number of chromosomes, and in daughter cells, this is known as mother cell and these are known as daughter cell. Daughter cell has the same number of chromosomes. Whereas in meiosis, 4, 2, 2, 4, 4, and then the result is 2. Meiosis take place only at certain time in an organism life cycle. My dear student, meiosis only takes place in the gamete formation in our sex organs, like in males, in testes for the production of sperm, and in female, in ovaries for the production of ovum. Right? So where we can find meiosis? Only in the reproductive organs. Meiosis involved in sexual reproduction, whereas mitosis involved in asexual reproduction. Mitosis will not lead to any kind of variation, whereas meiosis will lead to variation because it is going to give genetically unique cell. Thank you. Here we are ending our chapter. Thanks for watching.